Hey everyone, welcome to the live stream. My name's Brett Baker, and we just reached bottom off the coast of Uruguay. We are at about close to 1,000 meters. We're going to explore an area that hasn't been explored before using this the ROV Sebastian. Our primary objective will be to collect sediments, and we're gonna try and we're trying to look for these microbes that are named after Asgard, archaea. So they're named after Norse gods, and they're called Asgard archaea. Wow. We'll be monitoring, or we're watching your chat, so if you have any questions about anything, just let us know.
Yeah. Hi everyone, this is Valerie. I'm going to be answering questions in Spanish, and I'm going to be kind of translating everything here. Will you reach, estamos llegando a los 1000 metros. 1000 metros aproximadamente de profundidad. Estamos buscando sedimentos, en específico unas arqueas que se llaman Asgard arqueas. Y estamos muy emocionados porque hemos visto bastantes organismos muy interesantes. Un calamar fresa que se llama. Y bueno, ahora estamos observando el fondo. Este es un área que es completamente, que no se ha explorado antes. Y bueno, voy a estar contestando sus preguntas por aquí. Gracias por estar aquí. Jacob nos está explicando. Jacob, you want to go ahead and?
Sure.
Talk to them.
Estos animales gelatinos son más o menos blancos. Hay muchos y muchos. Son gelatinos de pene, o tenóforos. Son un grupo de animal que originalmente son pelágicos, viven en el columno de agua, pero esta especie y sus relativos viven abajo en el suelo del mar. Y algunos de los animales más profundos en el mundo son miembros de este grupo, los tenóforos.
Val, can you log the benthos
Yep.
tenóforos for me? Thanks.
Well, maybe we could get DNA from those.
That little polykey on the right.
Yeah. Yeah, it's a lot.
What's the species we're looking at? We're looking at different species, so people
these-
are asking
Yeah.
qué especies estamos viendo.
Los,
Or you can say it.
las estrellas son de un grupo de estrellas que en inglés se llaman brittle stars, en latín ophiuroides, ophioids, ophioidos,
Yeah.
y comen el detrito del suelo del mar, las estrellas. Este animal gelatinoso es un tenóforo o gelatina de pene y es de un grupo especial que viven en el suelo, en el fondo. La mayoría de tenóforos viven arriba en el columno de agua y a la izquierda, abajo, hay un gusano segmentado, poliquete. Poliqueto. Mucha diversidad.
Do you guys want the scanning lasers for some of this too?
Oh, lasers on the Singapore lasers.
People is complimenting your Spanish and they say that you're very good and that they can understand everything you're saying. So yeah.
Muchas gracias. Todo es latín. Es bueno para la ciencia.
Yeah.
Ophioido.
Ophiuros.
Ophiuros.
The lasers are 10
Yeah.
centimeters.
Oh my god.
Well, gracias.
Yeah, it's really small.
Yeah, they're tiny.
Okay, you happy? Move
Yeah.
on?
Yeah, let's move on.
My grad
Let's explore.
self is losing my mind. Yeah.
Try and get a good coverage of all the light. Too much of ISO. Do the iris before we do the ISO. Make sure the iris is all the way open. And then if you need to.
Sí, estamos viendo muchas estrellas de mar. La gente está diciendo sueño estrellado. And yeah, people complimenting your Spanish. That your Spanish is very good. They're asking if the colors determine the genera or that has nothing to do with that.
I don't think so. So as far as the animals we're looking at, the stars, the ophioids, the ophioros, have separate genders like we do. And I'm pretty sure that the color, at least in other sea stars I've seen, is just
It's
a
not-
polymorphism. It's just like how humans come in different
Yeah.
colors. The ctenophores, almost all of them, almost certainly
I'll zoom in a little
these,
bit as well.
are
It doesn't seem like there's.
they're hermaphroditic. So every animal is both male and female,
Okay.
Yeah.
and they all appear to be the same color
Yeah.
here.
Estamos en las costas uruguayas
You can describe
a casi 1000 metros de profundidad.
Yeah. Yeah, I'm sort of waiting. Let's move up and see if we can maybe. I want to get a sense of things before we do any cores, I guess, too.
Estamos explorando un poco antes de que empecemos a obtener los cores. Hoy en la mañana fue nuestro primer
Runaway.
dive y tuvimos muchos sedimentos.
But I would say we're like very light for the next dive. Like very
Too
light.
fast, I can't tell what that was.
Sí, prácticamente estas aguas que están sin explorar.
So we started around, I think I said we started around 1,000 meters, and our plan is to go upslope. So we're just off the continental shelf. Our last dive was on the continental shelf, so we only went to about 50 meters, and we just went off the shelf. And so it's pretty deep now. What we're going to do is sort of weave our way up the shelf a little bit and just see what we find.
Recuerdan que todos esos animales
Yeah, we'll just
son
kinda snake our way.
descendidos
getting them too
de
loose,
un,
because the idea is to try and
un
skirt around this
viejo ancestro
So
de Asgard arqueo.
only once we're really like
Ya.
getting a bit away.
Sí, lo que Jacob está recordándonos es que todos los organismos que estamos viendo ahora, todos los organismos eucariontes, todo lo que podemos ver con nuestros ojos, la diversidad que encontramos en el fondo del mar, así como la diversidad que vemos en nuestro planeta, proviene de un evento que se llama eucariogénesis, en donde las arqueas Asgard se unieron con una bacteria que al final formó las mitocondrias, y este evento evolutivo fue muy importante porque dio origen a la complejidad celular y a la complejidad de todos los organismos multicelulares y todos los organismos que estamos viendo, incluyéndonos a nosotros. Entonces estamos en la misión de encontrar estos microorganismos que se identificaron hace aproximadamente 10 años en el mar Atlántico Norte, en unas ventilas hidrotermales. Y a partir de ahí, estas ventilas hidrotermales tenían forma del castillo tenían forma del casco de Loki. Y a partir de ahí se empezó una nomenclatura utilizando los dioses griegos. Y este grupo de microorganismos se denominaron Asgard. Entonces, a partir de ahí los hemos identificado y encontrado en muchas partes, no nada más en ventilas hidrotermales y en ambientes extremos, sino también en el suelo. Y ahora sabemos que los descendientes directos que dieron lugar a la complejidad celular se encuentran también en el océano y en las costas de sitios donde hay bastante materia orgánica, como es el Río de la Plata. Entonces, estamos en búsqueda de estos microorganismos y, bueno, recordando de dónde venimos todos nosotros, incluyendo nosotros los humanos. La complejidad que vemos surgió a partir de la vida microscópica.
That's an octopus hole.
Is that, yeah, that's what I was wondering. Yeah, octopus hole.
Octopus hole, biological.
Yeah, biological observation. Right, those are crab shells.
Yeah.
At some point, our plan is we have the ability to acquire 18 sediment cores. So we have plastic tubes that will be filled with mud that will be
wait, maybe it's not an octopus
extracting
hole.
deep.
Those crabs are alive.
Oh yeah.
Yeah, something else hole. Yeah. So, are you looking for a space that's clear of ophiurites?
Yeah, I just want to get a sense for things a little bit, yeah, before we core in, maybe get away so we don't get. Whoa.
People are asking
You're happy, Brett. We
what
just started
is
moving
the natural
west
predator of the stars.
west,
Oh.
northwest.
Yep.
See the
Good
Falkor
question.
waypoint
Yep.
there? Yeah,
I
look at
don't-
that.
Yep.
I
We'll
don't
just
know,
kind of
and
beyond
probably
there.
not
Yeah,
much.
great.
All I can say is there's not very much meat on them. No es mucha carne.
No hay mucha carne. Nadie
Um,
se los come.
They're decomposers. So they're kind of at the end of the
Están
food
en
chain.
al final de la cadena trófica, entonces se encargan de degradar compuestos que están básicamente caen en el fondo del mar.
Yeah, looks like trash, maybe.
Um,
What was it excuse? When I know stuff.
Wow. A lot of them.
Not
yeah.
I think they
seeing
just.
what everyone else is seeing sometimes.
Endless.
Yeah.
La gente está preguntando dónde estamos para aquellos que se acaban de conectar. Estamos en la costa de Uruguay a casi 1000 metros de profundidad, un poco fuera de la sí, a 1000 metros de profundidad. Estamos aquí registrando todas las especies que estamos viendo. They're asking why there's so many and
Yeah.
what's approximately their size.
Well, how far apart are our lasers?
The lasers are 10 centimeters apart.
Yeah, so they're
Like
like
5.
at most 10 centimeters.
Yeah.
Arms stretched out.
Yeah, there is one there that looks like a 10-centimeter animal.
Yeah.
So the two green points in the frame are parallel lasers that are projecting off of the vehicle, and they let us measure things. They're 10 centimeters apart, so that gives you an idea of size.
Para aquellos que preguntaron de qué tamaño son las estrellas de mar, si pueden ver en la pantalla hay 2 puntos. Esos 2 puntos tienen 10 centímetros, entonces más o menos nos da una idea de qué tamaño tienen las cosas. Pueden haber de 5 centímetros, de 10 centímetros. Entonces esos son los láseres que están proyectados desde el submarino.
Mm-hmm.
Apparently, some fishes and crabs and things occasionally eat brittle stars.
Some,
But
yeah,
crabs will eat anything
yeah.
if they're hungry enough. But their arms break off very easily.
Yeah.
And they regenerate them just like any other sea
Yeah.
star. It's good for escaping. They can actually detach them at will.
Oh,
Yeah. Which is different than most other sea stars.
no, para aquellos que preguntan si vamos a colectar especies, no vamos a colectar especies biológicas, estamos enfocados en los microorganismos que viven en los sedimentos. Ahora estamos explorando un poco para ver en dónde podemos poner estos los cores para poder obtener el sedimento, solo que como podemos ver está lleno de estrellas. Aquí podemos
Wow.
ver un cangrejo.
Oh yeah.
Oh boy. Getting friendly.
Yeah. Oh my God, how do you...
This is going to be. Wait. Oh
Where
my
is
gosh,
it?
we're missing the biggest. Put it under other. Sorry, we're missing the biggest phylum.
Yeah.
You can put crab. That's embarrassing. Sorry.
Estos cangrejos están... Quieren tener un poco de
I feel like we
privacidad.
interrupted something here.
Un don cangrejo. Dos. Se van.
Later.
Speaking of new species, by the way, there is no way that those ctenophores are a described species,
Yeah.
or even close to one.
Yeah. Okay.
So
So new.
Anybody want to suggest a name for the ctenophore species we just found?
Well, it's a little bit more involved than that.
Yeah.
But we can come up with a nice Spanish or English name.
Yeah.
Oh, somebody just jumped up from the bottom there.
Where was that?
On the left. Yeah. There it is. A red thing.
Oh,
I think it's
Oh.
gonna be in
Probably a
the-
shrimp.
now.
Yeah.
Yeah, it's a.
it's a tiny shrimp.
Yeah, it's a shrimp, I think.
Un cangrejito. No, un camarón.
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 Camaronito.
Es que me quedé pensando en los cangrejos.
Creo
Sí.
que es un camarón sergéstido. De esa familia.
¿Cuál es?
I'll see. I mean, other shrimp. Let me. I'll
Yeah,
try to
we
fix
need
the- I'll
to
try
we need
fix
to
this.
fix. Yeah.
Huh?
What?
Los crustáceos. Yeah, I was like, there is something missing,
There's
or
that
I'm
one.
like,
It
they
is
changed the
only the biggest
the,
animal phylum on
the
the planet.
phylum.
Yeah.
Hola desde Argentina. So why are the shrimp so very, very red?
That is an interesting question. So, is this asked in English or Spanish?
Yeah, we'll just leave it.
In English.
Yeah, so red in the deep sea is a really interesting recurring pattern. Naturally down here, there's basically no light, especially at night, but even during the day, we're so deep that the sunlight doesn't get here. We're just putting our white lights on it from the vehicle. And so the colors that we see with the ROV are pretty unnatural compared to the colors that animals that actually live down there would be seeing. As you move downward in the water column, we all know that water appears blue, and it's because it scatters blue light. But water is also really good at absorbing warmer colors of light. So as you descend, the first color that goes away is red, and then you lose orange, and then you lose yellow, and so on down the rainbow. That means that anything that is red colored down here is actually going to show up black to the animals around it, assuming there's not an ROV around messing everything up. So if you are a shrimp with a red body, you're going to be basically invisible to your neighbors. Some of these animals, like that shrimp, you'll notice are partly clear, but only the stomach is red. And it's thought that that red pigment is there to block out food that they might eat that actually lights up. So a lot of organisms down here, about half the animals you find living on the seafloor can actually make their own light. And if you eat something that's blinking, that's a really good way to accidentally attract one of your predators. So that's why we think stomachs are red.
Oh, it just keeps going.
Okay, so now you have to say that in a shorter version in Spanish. They say, yeah, that's pretty cool, but I don't speak Spanish, some of them, so.
Okay, voy
So
voy a
just like a
tratar.
little, like a shorter version of
sí.
that.
Más simple, más corto.
Más corto.
Y creo que la anotación es arreglado.
No sé si están viendo en la. No, I,
Sí, pero
for that one, but that's like it's gone.
I'm just saying
Yeah.
it's there.
Okay.
Okay.
Oh, you just did it. Okay, awesome.
muchos animales en el mar profundo tienen color rojo y eso creemos es porque naturalmente no hay luz rojo en el mar profundo. Ahorita tenemos luces en el ROV que son blancos y por eso podemos ver cosas rojas, pero naturalmente las cosas rojas en el fondo del mar parecen negro o invisible a los otros animales. Y por eso muchos de los animales tienen pigmentos que son rojos y algunas tienen específicamente estómagos que son rojos porque esos oscuran
Interesting way of moving.
comidas que los animales pueden comer que son bioluminescentes y el pigmento oscura la luz bioluminescente que puede ser dañoso para un predador.
Peligroso.
Sí, peligroso porque puede atratar predadores.
People are very grateful and they say we appreciate you all speaking Spanish. So
Muchas
yeah.
gracias para su paciencia. That is es una medusa. Creo que sea una medusa de las aguas más de las aguas más
¿Someras?
someras. Ujú.
Which
Pero creo que es muerto.
yeah.
That. Ujú.
Very good. Learning.
Yeah. Ujú. Exactamente. Y cosas como esta medusa muerta es, ¿cómo se dice? Es toda la comida para animales como los ofiuros. Comen solo cosas muertas.
So, para recordarles un poco, estamos, para los que se acaban de conectar, estamos a 1000 metros. People really appreciate you speaking Spanish. So, yeah, thank you. Thank you for the effort. Para recordarles, estamos a 1000 metros. Aquí no llega la luz del sol. La luz del sol llega aproximadamente a los 200 metros, ya no hay luz. Entonces estamos a unas profundidades donde estamos en total oscuridad. La luz que vemos viene del submarino, del Sebastian. Entonces, simplemente para recordar, estamos viendo animales que se encargan de descomponer materia orgánica y todo lo que viene de la superficie, ya sean animales muertos.
Oh, never.
¿Cuál?
No, it's a fish.
Animales muertos, materia orgánica, todo lo que se está descomponiendo, estos animales básicamente lo comen y después al final no lo estamos viendo, pero los microorganismos también se encargan de degradar toda esta materia orgánica y de reciclarla. Los sedimentos marinos guardan el carbono que se fija en la superficie del océano, el CO2 que está en la atmósfera, se fija en la superficie del mar, produciendo materia orgánica. Esta materia orgánica se recicla y al final de todo, en los sedimentos que estamos viendo aquí, los microorganismos se encargan de reciclar todo esto y guardar ese carbón en forma de metano. Los sedimentos marinos son el reservorio más grande de carbono en el planeta en forma de metano. Y eso no lo podemos ver como vemos estas estrellitas de mar, pero simplemente para recordar que esos procesos están pasando y son básicamente un engranaje que permite que el ciclaje de nutrientes fluya en el océano y en la atmósfera.
So, if we collect 9 cores from a site, we will be able to give enough to everybody that needs cores, or if we split it, what if we split it 3 sites, get 6 cores? Will everybody get enough? So
I think
Val
if-
and I are just discussing, we have like 25 scientists on board that are all doing different things
I will go forward of the bridge for 1 minute.
with samples. So there's people that are doing geochemistry, there's people doing DNA extractions.
Not off bridge, just the other side of the bridge. So if you call me, it will take me time to answer.
My research team is here collecting mud, and we're going to extract DNA and RNA to look at the diversity and reconstruct genomes of the microbes that live here. And then there's other groups that are going to try and culture these Asgard archaea, so essentially grow them in a laboratory. What we're trying to do is understand where the Asgard archaea are our closest relatives on the tree of life. So we're trying to explore their diversity in nature to get a better understanding of essentially the shape of the tree of life and where we came from on it. Yeah, just really cool. What do you think? Can we do 3 sites maybe?
I think if we do 2, 6, and 6, that will be plenty.
2, 6,
9
and 6.
and 9.
With 18, it's a lot.
Yeah.
So if we do between 12 and 15, it will be more than enough.
Yeah, so we'll do that.
Oh wow.
Is that?
Cycle of life. A dead crab, but alive something else.
Estamos viendo un cangrejito muerto, pero estamos tratando de ver qué es lo que está viviendo.
Y una colonia entera de
Una
algo
colonia
otro.
entera de otra cosa que no podemos identificar. Maggie says hi. She's also watching.
Maddie?
Maggie Langway.
Oh. Hola, Maggie.
Oh, I know Maggie.
Yeah,
Yeah,
everybody
yeah,
says
yeah.
Maggie.
Yeah.
Hi, Maggie.
She was on, yeah.
Welcome back.
Switch out.
Yeah.
Yeah.
Huh.
What is that?
They look like worms. They're polychaetes.
Yeah, they look like worms, right?
Yeah.
You guys don't any Cusco? No. At the moment.
Not yet.
We are not looking for the origin of life. We are looking for the origin of complex life. We're looking at understanding once life originated, and then 2 billion years after that, what happened when the ancestors of the modern Asgard archaea merged with a bacteria and gave rise to a first eukaryote. So we're trying to understand the modern archaea to kind of model what we could have happened in that moment, in the origin of complex life.
I don't know if you guys can best go away somewhere. We're trying to figure out how to.
People are asking if those are fungi.
I think I was wondering the same thing. I think based on our zoom in that they're actually tiny worms, but you really have to get in
Yeah.
close.
Are
you going to?
If you look at some of the very small ones, they're crawling around.
Yeah.
Good.
Did you do that?
Yeah. Just for you.
Just for you, Jimbo.
2 mics around.
You can ask them to pull in further if they can, if you want.
You know how they work.
ROV bridge back into the
Yeah, so see how they're segmented.
Yeah,
And
they're
they're crawling around.
yeah, those are worms.
We should continue on.
Okay.
Or do
Yeah.
you guys want to look at? Yeah.
Yeah.
Yeah, I think we can move on.
Yeah, we can move on.
Yep.
Anyway, I'll go ahead.
So my only
Hi, Angie, my wife. I love you. Hey, Silas. How's it going? My son's online too.
Sí, es un cangrejo. Lo que vimos fue un cangrejo en descomposición y había varias especies que se estaban alimentando de él, gusanos y las estrellitas de mar que vimos. Entonces
Well,
son.
I was kind of hoping we get out of these stars and do some sediment coring, but no, it doesn't look like it's going to happen. So
Yeah, maybe like a clear area over there. Like,
yeah, you see a clear area?
Like a little bit.
Yeah.
Yeah.
I don't know.
She's saying.
I see little spots.
Yeah.
They'll
Like-
I think they'll run away after you land.
Yeah, I think they are going to run away. They do want to get away from us. So
Not nice.
yeah. So why don't we go ahead and start sampling?
Yeah.
Before we land, can you trigger 3 Niskins? So what we're gonna do now is there's bottles that are open on the side of this the ROV, and what's gonna happen is they're opened on both ends. So we're just gonna essentially trigger them so that the ends close, and then we'll have the water that we can bring back up and do some analysis of.
What else do
Can you get like as close to the bottom as you can without hitting? Yeah,
Before we did, we were about 1.5 meters
about
altitude.
1.5 meters. Okay, yeah.
I'm gonna try to get some of this mud off of this so that we're not gonna get that in there.
Yeah, that would be great.
That's the mud off the bottom.
Great. Yep. Perfect.
So right now we're 1.7 meters off. Alright.
They're asking exactly where we are. They know that we are in
Yeah.
Uruguay, but they want to know where.
We're
I'll put a map up then.
Yeah,
sure.
if you
Yeah,
can
we'll put a map up of where we are. We're like.
Para los que se acaban de conectar, vamos a poner el mapa para que vean exactamente dónde
Okay,
estamos.
great. She triggered 3 Niskins.
3?
Mark it. Yep.
No,
Oh, shit.
sample. There you are.
Alright, and somebody sit down and you guys
Yeah. Go ahead and sit it down.
How many?
We'll do
8, 6,
6.
3.
Ready to quarter starboard.
Remember, there's no hurry.
What's stuff for
Got it.
starboard?
Until we know.
No. The Niskins are triggered. So the Niskins are done. I'm just saying that since you.
No,
clicked
no,
it.
but just 3. We don't know which ones.
1, 2, and 3.
1, 2, and 3?
Yeah. Niskins have to be triggered in order.
no.
You can't do multiple, so you'll have to do multiple annotations. Does that make sense?
Yeah.
Yeah. So you'll.
What's up, Jimbo?
What's it, sorry?
How you doing? What'd you have?
I just put it aside right now.
That?
Next to the-
Yeah.
You guys want us to start in order, like 1 through.
Yeah, sure. Yeah, that'd be
Okay,
great.
yeah, so-
Yeah.
And just you can go ahead and push them all in first, and then we'll pull them.
So the only request I have coring wise is
Yeah.
I'm pretty late in the cycle. I'm number 11.
Okay.
And I just want to make sure that I am next to Hyde's core.
Okay.
Like, ideally we're.
Yeah, but
co-located.
we decide that once we see
After
which
the
one.
fact.
We can
Yeah.
do that afterwards.
Yeah.
Yeah.
Okay.
Yeah.
Well, maybe if you get a nice deep core and you feel like picking up 11 and sticking it next to it.
Okay. That's you.
Yeah, yeah,
You're
yeah.
11. Okay.
Yeah.
And hides is just any one that we like, or is it a specific one?
The nicest one.
Yeah.
Yeah, just any
So you're
any
here.
core.
You get whatever you want.
Any core that is next to 11.
They're all going to be.
So
close
we're
together.
just discussing now the sampling and who's going to get what. Just to give y'all some background, we're landing now, so we're gonna start using the manipulators to pull some sediment cores.
Brad?
Yes.
Question about whether you're going to look for microplastics in these samples.
Yeah, that's a good question about the microplastics. Someone asked if we're gonna look for microplastics in the samples. Probably. We don't have anyone on the research expedition that's going to be focusing on it, although I have some collaborators at University of Texas, where my lab is, where we've looked at, we've actually looked for microbes that degrade microplastics, and others that look at the composition of microplastics, and we've actually found from hydrothermal vents, we've pulled some new enzymes that degrade plastics that are pretty cool. So yeah, I'm going to be giving samples to people that will be looking for new enzymes to degrade microplastics.
Oh, Giovanni. Hola, Geo. He said, good luck finding archaea. Let's try to isolate methanogenic archaea too.
Doesn't matter. Okay, so we want to avoid
Oh.
the biology though, so if you can tell
Hola,
Geo.
a little spot that's kind of
Yep.
bare.
Te saluda también Jacob.
Yep.
Enjoy it.
Move away.
So you don't have to put them in order. I think we're just gonna see how they look. So just Jacob's number 11. That's
Yeah.
the tricky one.
Yeah. Mostly, I'm just hoping that if you're gonna core 2 sites, I'm
Yeah, the
hoping-
one that is next to it.
I'm
yeah.
hoping that I can and I can talk with Emily about this, but like we would love to be able to cross.
Yeah. Tenemos en la
Yeah.
expedición
Do as much as you
científicos uruguayos también. Ellos están haciendo CTD. Uy,
That's good. Beautiful.
You want to leave it or take it out?
Leave it.
Okay.
Wow.
saludos a ti también, Geo.
Yeah, just like you pull on it, disturbs the top of it. Yeah.
At least
So-
two.
Yeah.
I think it might be more than that.
Yeah, we have
Yeah, if you.
our science
yes,
party
only do it once it's
has
back on the vehicle.
researchers from
If you want, you
so
can,
myself from University of Texas. We have University of Vienna. So
if you
Val
want to.
Dianda is the other person that's been narrating this. We have people from Harvard, Cambridge, in the UK, obviously. Charles University in the Czech Republic. And of course, we have some great collaborators from here in Uruguay. It's been a long day trying to think of everybody.
Rhode
And
Island.
University of California, San Diego. I think that's.
Are we going nationalities or institutions?
Yeah, I didn't do. These are institutions. that people work at.
Institutions, yeah.
Nationalities is much more complicated.
Yeah.
Yeah.
Because they said which countries are they from, and they said which
Yeah.
The exact thing was.
Uruguay, U.S., Czech Republic.
Bueno, aquí
Yep.
en el...
Yep.
There was a
Yep.
direct question, are there any Uruguayan scientists on board?
Oh yeah, of course.
Yeah, there-
yeah.
Hay científicos de Uruguay, solo que no están aquí en...
There's
Control.
there's 5 Uruguayan.
But it's like the ship's crew, you know, we've got about at least a dozen
Oh,
different nationalities. on the
Yeah,
ship's crew, so-
this is just the science party I'm talking about. So the ship's crew has, yeah.
But I was talking about the science party, and so you've got a fair few nationalities represented there.
Yep.
Bueno, hay 2 mexicanos aquí ahora.
We're doing our
Boy.
best to avoid the animals while we try and get some mud. Yeah. Of course. Yeah.
So you want 6 plus 11 or 5 plus 11? 6 normals and then 11, or do you want 5 normals?
What was the question?
Do we want 6? Wait.
6 in one
Oh,
side?
plus 11.
Yeah, number 11.
Number 11.
Yeah, yeah.
So
Yeah.
6 and then number 11? Or 5?
5 and then number 11.
Okay.
Yep.
So 6
Yep.
total,
Yeah.
Yeah.
including number 11.
Yeah.
Well, my core is a wash. I take it home. So if you want to have material from 6 cores to share around,
Oh, okay.
yeah.
So then let's do 6 plus 11.
Gotcha.
Yeah.
But this looks like beautiful sediment. So
It does, yeah.
for including
These will be
me.
great. These will be great samples.
That's a lot of pressure going. You want that little mark right there? Yeah.
That fluffy layer on the top that sort of floats away, looks like smoke, is referred to as the nephloid layer. Yeah, nephloid. Yep.
Not careful. We're going to core one of those
There you go.
cores.
Yeah.
A word a normal person would never need to know.
¿Por qué el lugar donde están es interesante para buscar Asgard, arquía y cómo deciden los sitios de muestreo? Thank you for that question, Lucas. Hay bastantes razones por las cuales es interesante explorar el mar uruguayo. Básicamente encontramos Asgard arquea en varios lugares en el mundo. Al principio solamente se habían descrito en ventilas hidrotermales en el mar Atlántico. pero haciendo una búsqueda exhaustiva en el laboratorio de Brett encontramos que estas arqueas se encuentran en básicamente en todos los lugares que hemos muestrado, a una profundidad de secuenciación más grande. Entonces, en el mar Bohai, en China, encontramos una gran cantidad de Asgard, específicamente Hodarkea. Estas Hodarkeas son el linaje que es más cercano evolutivamente a los eucariontes y se encuentran en lugares costeros con una gran cantidad de materia orgánica. Entonces este mares uruguayos es el sitio ideal para poder encontrarlos. Muchas gracias.
I see someone asked how long the dives are in this cruise. So our first station, I lost track of time. I think it was yesterday, was only 50 meters, so it was really quick dive. So one of the longest parts of doing sampling with the ROV is that it takes time for the ROV to get to the seafloor to get sediments, right? So it's just like roughly 1,000 meters an hour. So we're at 1,000 meters now. So it took, I don't know, it probably took a little more than an hour because we stopped and we saw some things along the way. So this dive, I don't know. We'll see how long it takes. We're already doing the first sampling, so, but eventually we'll get further off the shelf and we'll be collecting samples from 3,000 meters. So that's essentially 6 hours just up and down, and then other operations, deploying the ROV and recovering it from the ship. So some dives can be 15 hours, upwards of. It all depends where we are.
They are asking if we're going to be doing metagenomics. En español van a hacer metagenómica de estas bacterias o cómo las identifican. Son arqueas y sí vamos a estar haciendo secuenciación a bordo del barco. Esto básicamente vamos a extraer el DNA y secuenciar a bordo para poderlas identificar en tiempo real. Además de metagenómica, con estos sedimentos vamos a hacer muchas cosas, cultivarlos. Se va a, también el equipo uruguayo está midiendo isótopos, está viendo materia orgánica, están haciendo bastantes cosas con ellos, se están haciendo cultivos, etcétera. Entonces vamos a hacer un esfuerzo de colaboración interdisciplinaria para poder atacar o utilizar diferentes disciplinas para poder encontrar a estas arqueas. Muchas gracias, Paola, por tu pregunta.
You want to start logging these in the...
Once they are in the quiver, just let me know which one and I will start.
When it goes in the quiver, I'll tell you which core number it is?
Yeah.
Okay.
Whether the bond to face the camera or the opposite, doesn't matter.
Right there. Is that close enough?
Yeah.
Alright.
Yeah.
This is not going to be disturbed by the when you just put it in.
Yeah.
It's not gonna every time.
Yeah. It's pretty homogeneous here, so I don't think it's going to be that much different. A little farther away. He escaped. Sure. Yeah, might as well. That's where you are. Yep. So we covered
Vale.
11 first. Yep.
Okay.
So this will be PC1.
Yeah.
Yep.
PC
No.
11.
Wait.
PC 11.
PC 1-1.
Oh, okay. Thank you for clarifying that. Oh, man.
Okay, that's a nice looking core.
What's your which we have say the tension is right
Tension is 580 kilograms.
now? Okay, no, that's about what it says here. It's dropping down now.
Could be currents going on it. You can give it a little bit and then it would slacken off.
It says 570.
It was over 500 kilograms. I was pulling it in.
Yeah.
Back with that.
La razón por la cual sacamos las muestras en el mismo lugar es porque queremos coordinar los metadatos, queremos coordinar, utilizarlo como réplicas para diferentes cosas. Unos cores van a ser utilizados para cultivo, otros cores van a ser utilizados para secuenciación a bordo del Falkor, radioisótopos, etcétera. Entonces queremos tener todos los sedimentos más o menos en el mismo sitio para poder utilizar estos metadatos. Okay.
Yep, log it. PC11.
Are we still expecting recovery at 10 a.m.?
That's
No.
the last I heard.
Take that one.
Yeah. So what are you thinking?
Los pilotos, aquí tenemos a Joel de Veracruz y Chris. Mandan saludos a los pilotos, al ROV team.
Something more like 3 probably.
I'm going to bed.
Yeah, you should go to bed.
Unless you need me.
Yep. No, you're good. Thank you very much.
But 4 hours.
Yep. I'm not gonna. I'm gonna do more multi-beam after this.
Okay.
So it's not like I don't. Laura is going to be taking over whenever she's done filtering. Great. And I want to adjust the schedule.
Okay.
So I'm not expecting things up tremendously because everyone's going to be bad. I can't.
Can I just take
Just so it
one
doesn't
picture
affect
of
the
that?
Well,
Yeah, just a close-up of the
The
cores. I
only-
just want to see how close they are.
The only
Thank
concern
you,
in my mind
Joel.
is I don't know how much people you'll know better if you do this by all.
Yeah, I just took a picture of like this layout just so I can see how far each one is, but I took it. Thank you.
I don't know how much people will care about sample freshness. It's like if we can just pull them off the vehicle and park them in the cold room, that's well and good. People will wake up and deal with their samples.
Yeah. that's the problem.
But if people really want their samples fresh off the thing, then
Yeah, exactly.
you know.
We'll get them in the cold room.
Yeah,
Yeah.
Yeah, you think that's they're stable in the cold room?
Yeah.
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 Okay.
Yeah.
They're fine. I was thinking more downstream stuff.
Well, it's if they're stable to sit around for the downstream stuff, then it would seem like the upstream stuff would be the bottleneck.
No,
No.
I meant like with the next.
Oh, successive ops.
My mind, yeah.
Yeah,
Ops,
yeah,
sorry,
yeah.
I should have been more clear.
No,
Yeah,
it makes
yeah,
sense.
yeah.
Um,
Which one is this one?
This is I don't know.
Push core.
okay.
We'll see once it's in there, probably.
Yeah.
So are you saying I don't need to be up at 3 to help unload the vehicle?
I am. You're fine. Get some
Okay.
rest. Yeah.
Likewise.
Thank you. Yeah.
And hopefully
Hi.
whomever is on watch, because they're already planning on being up at that time, can help
Exactly.
unload.
Yeah, and I'll stay out too. So it's cool. Yeah.
All right.
Thank
You
so
got the
much.
little one.
Yeah. Big help. Big help.
It's getting fun.
Yeah.
Noches.
We'll do our best to answer questions, but we're taking notes right now as we get samples, so just be patient with us.
Go ahead.
Number 3.
Sorry, Jimbo, I had the alarm at the same time. Say again. That's fine.
Yeah. 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 It's not a huge deal, but it would be, if we can correct it, would be great. Yeah. Yeah.
Number.
Right. Yeah. Okay. Yep. Great. Thanks.
Very nice place.
Yeah.
Yeah, those are
That's lovely.
very nice looking cores. This is number 1.
Yep.
Yes. Number 1.
Thank you. Gracias por su paciencia. Estamos tomando algunas notas mientras hacemos la recuperación de los push cores
202 kilometers.
y tratando de responder a todas sus preguntas. Gracias por sintonizar y por estar aquí.
Get all these.
Ujú.
Okay.
Jimbo. 202 kilometers. 202.
So this is number. I didn't see if it was number 6, right? I think it was number 6.
6. Yep.
Got it. Estamos a 200 kilómetros lejos de la costa de Uruguay, a 1000 metros de profundidad, recuperando los últimos push cores. La temperatura de la superficie es 3.3 centígrados.
There you go. Yes, sir.
Aren't any hydrophones on the ROV?
There are no hydrophones on the ROV at present now.
Number...
Yeah.
Is this number 4?
Yes.
Thank you.
Mm-hmm. Well done.
Number 2. Thank you.
Alright, anything else at this spot, or?
I don't think so. I think we can lift up.
Brett, they are asking what's the maximum depth we're gonna...
Yeah. On this expedition, we're not entirely sure what the maximum depth is going to be, partly because we're going places that haven't really been mapped that well. But if it's going to be roughly around 3,000 meters is the deepest planned site that we'll be going to. The site that we're at now, yeah, as you can see, is about 1,000 meters.
Sí, en esta expedición no sabemos, no tenemos muy claro, pero cuál es la profundidad máxima a la que vamos a llegar, pero tenemos calculado unos 3.000 metros, que va a ser la profundidad máxima aproximadamente. Ahora estamos a 1000 metros y vamos en búsqueda del segundo sitio donde vamos a muestrar los sedimentos.
Yeah, it's fine. That's good.
Great, so Lee, you have the weapon. Rich, Charlie.
Go ahead.
Yeah, new weapon, mate.
New weapon.
Thank you. So, that's the.
Yeah, we hadn't been listening to it, like we were at 10 or 50,000 under ISO. You did all that. I don't know how you saw it already.
Chris Elson.
No, just the Irish stuff.
Okay, we're lifted up and we're gonna go, gonna just head upslope a little bit. It's a pretty gradual slope. So just gonna look for another spot to get some more sediments. We got some really nice cores from that first site on this dive.
Yep. So, Geo.
Gio's asking.
Yeah.
Gio's asking if Asgards are aerobic. Hey Gio, how's it going? Glad you're here. That's one of the things we're aiming to figure out on this cruise is to test if Asgards can. So if you don't know, aerobic is using oxygen like us. So that's interesting because if they are our closest ancestors in the microbial world, then it's been suggested based on their genomes that they can use oxygen. So we're going to test, and we're doing some experiments on board and doing other things when we get home to see if we can prove that essentially.
Para traducirlo en español para para los demás, básicamente estas arqueas, a partir de sus genomas hemos deducido que tienen un metabolismo capaz de utilizar el oxígeno como aceptor de electrones, es decir, para poderlo respirar, una cadena de transporte de electrones. Y haciendo la reconstrucción del metabolismo del ancestro que dio origen a la vida compleja, el ancestro de las Hod arqueas, hemos visto que pudo haber tenido una cadena de transporte de electrones casi completa, lo cual significa que tuvo, además su metabolismo también indica que era un organismo mesófilo y heterotrófico, es decir, consumía azúcares y producía ATP de la misma forma que nosotros lo hacemos. Y a partir de las reconstrucciones de los metagenomas y de los genomas que hemos visto de esas arqueas que viven en ambientes como estos, también reconstruyendo estos genomas hemos visto que tienen la capacidad de utilizar oxígeno como último aceptor de electrones. Sin embargo, no las hemos podido cultivar ni las hemos podido ver. Entonces eso es lo que estamos tratando de hacer en esta expedición y estamos muy emocionados de poder utilizar diferentes disciplinas, colaboraciones con diferentes científicos de varias universidades para ver si podemos encontrarlas.
Arabi Bridge.
Go ahead, Rich.
Just to keep you in the loop, it's getting a bit windy up here, but everything's under control. Just an unexpected wind.
You're doing a great job, Eric. You're doing a great job.
If you're just joining us,
Thank you.
we're off the coast of Uruguay, and so it's an international team. We're looking for primarily looking. Our main focus of the cruise is to look for microbial life. These are Asgards that are related to us, but we're also doing other work, and so we're doing that in both the water column, in the water, and in the sediments. So we're using the ROV to get mud, obviously, in the sediments. And ever since we landed on this dive, it's been nothing but brittle stars everywhere.
Eric, you have a new waypoint.
Gracias a todos por sintonizar y por todas sus preguntas de la Universidad de Viena, y también soy exploradora de National Geographic, y estamos coordinando todo el muestreo de los sedimentos. Es muy emocionante, todos estamos muy contentos de recuperar estos sedimentos. Ha sido un trabajo en equipo. Y aquí están preguntando cómo es el tema de la respiración en las arqueas. Las arqueas es un dominio de la vida, entonces estamos hablando de la vida que conocemos en el planeta. Se divide en 3 dominios básicamente: los eucariontes, las bacterias y las arqueas. Como procariontes, las arqueas y las bacterias son organismos que tienen un metabolismo muy variado. Nosotros como eucariontes somos metabólicamente aburridos, como yo les llamo, porque respiramos y hacemos fotosíntesis. Básicamente consumimos y producimos azúcares. Las arqueas y las bacterias no. Ellos tienen una gran variedad de vías metabólicas para poder respirar diferentes compuestos inorgánicos, como el azufre, como el metano, y lo hacen a partir de una gran variedad de vías y rutas metabólicas. Hay algunas arqueas que son muy características de sitios que viven en ambientes extremos, como se descubrieron en ambientes extremos, se asociaron a ambientes extremos. Sin embargo, se han encontrado prácticamente en todos los ambientes, incluyendo en la superficie del océano, en el fondo del océano. Y bueno, el metabolismo es básicamente las reacciones que mantienen a cualquier ser vivo, que mantiene ese ser vivo, ¿no? Estamos básicamente explorando un poco hasta encontrar el siguiente sitio. Hi Angie, nice to see your comment. Hope you're doing well. Saludos a Austin. This is another squid. Can we just zoom in a little bit?
Heck. What's that? I am a microbiologist. Yeah, both of us are microbiologists, so.
Sí, el encargado de la descripción de los organismos eucariontes se tuvo que ir, pero sí, nosotros somos microbiólogos y desafortunadamente no podemos ver los microorganismos a simple vista. Tenemos que utilizar técnicas para poder cultivarlos, analizar su metabolismo, sus genomas. Así que no podemos darles el nombre científico de todas las especies que estamos viendo, pero estamos tratando de utilizar a los expertos para que nos digan los nombres científicos.
Take it off. Yeah. Brought one of those up the other day on accident. It's horrifying.
Sí, para los que se acaban de conectar, sí, todos
the
estos
fishy
son
thing?
ofiuras, son
Oh my
estrellas de mar. Está
Wow.
It's underneath the pressure.
metros y metros y metros
Yikes.
llenos de estrellas. Es una noche estrellada. Están preguntando qué tan rápido baja el submarino. Aproximadamente 24 metros cada minuto. Así que para llegar a los 3000
Yeah,
metros, aproximadamente 2 horas.
Woke him up.
Which they went with the male?
I don't know.
I have no idea. Really calamari.
Yeah, calamari.
Uh-oh. In and out.
I kind of hope so.
Estamos a 200 metros, 200 kilómetros, perdón, de la
Yeah.
costa de Uruguay, a 1000 metros de profundidad. Chief
Hey,
Scientist Brett Baker y
Welcome.
acaba de llegar Pavel de República
We,
Checa y yo, Valerie
I
de
can,
Anda,
show you what's going on
de
here.
científica en la Universidad de Viena.
Let you take over in a bit.
So what do I do?
Yeah, I'll show you. We can go over here.
You want me to select a site to start push course on the other one, or you want to do it and
Well,
fire, then it's
I'm
gonna...
not leaving yet.
Oh, okay, okay,
Just-
okay, okay.
Yeah, I'm just showing them. So this is our simply basically our notes. But Val's gonna be logging in on the computer, so this is just kind of a backup. But essentially, you can ignore this. So my handwriting is really sloppy, but P
Hello guys, here is Pavel. So I'm here with my colleague Robert. We are from Prague, Czech Republic. I guess some of you may know where Czech Republic is. It's a pretty small country. We are in landlocked country, so no sea. So this is actually my first time on such a big ocean trip. So what we are doing at this cruise is so we are molecular parasitologists, meaning that we just we spend most of our time in the lab studying proteins and other molecules which somehow associated with the parasitic lifestyle of some of the pathogens, and we specialize in studying parasitic protists and also free living protists. So at this cruise we are trying to collect these invisible cell organisms. So these are organisms which are composed of only a single cell, but they can do everything just on its own. So they have the same cellular structure as our body. So these are not prokaryotes, these are not Asgards, neither bacteria. So these are eukaryotic cells. And some of these, so we believe that these sediments we are just watching now contain some of the, some of these protists, these unicellular eukaryotes, which can tell us something about the first eukaryotic organism on our planet. So of course we don't know what we are going to see, and we will see only later when we analyze the material from the sediment. So we can say that the first eukaryotic cell was a protist, meaning that it had a nucleus, it had a mitochondrion and other cellular organelles, but it was a single cell. Many of these protists also live inside other organisms, and this is also possible thing which we can study here. So we can take, we can analyze, interior of other animals, or, but this is not the main purpose of this trip. So we will be just analyzing the sediment and whatever it freely lives on the surface layer of what we see. It's also possible that some of these things live as spores. So many of these organisms actually don't look alive. So they might be in a dormant stage. But when we take the sediment to the medium, so we have several different media, meaning like growth media. So these are like soups made of different nutrients, which we have in different tubes. What we do, we'll just take pieces of this sediment, put it in the different tubes of different media, and we hope that this media will just support life of some of these protists. So they will actually have better conditions than they have in the sediment, and they will multiply in our media. And possibly, if we succeed, then we can establish cultures of these organisms, and then we can study their cellular structure in a more detail. So we can go to the microscope. We can analyze the cellular structure of these organisms. We can isolate organelles. Of course, we can isolate DNA, but this is something which we can do even from the raw material from the sediment. But if we want to establish like a model which we can take into the laboratory, then we need to establish some cultures. what is important in these environments is that the surface layer of this environment is rich in oxygen, so it's a regular. So we think that these organisms which live in the first couple of centimeters will consume oxygen, will use oxygen for its life. It produce energy using oxygen, so this is called respiration. But in the deeper layers, but even like starting from 10 centimeters below the surface of the sediment, there is very few oxygen, so we can expect like really anaerobic lifestyle. So this is also something very special. So we can see organisms which don't use oxygen,
Rich, new waypoint.
and which have a completely different metabolism. So we can find organisms having completely different cell metabolism across the sediment.
Rich, new waypoint.
Okay, waypoint.
Hola a todos los que nos sintonizan. Estamos tratando de contestar todas sus preguntas. Sí, estamos en Uruguay. Hay científicos uruguayos a bordo de la expedición. Ellos ahora están trabajando con el agua. Hay 5 uruguayos a bordo, pero también están muchos más que van a analizar las muestras y los sedimentos y el agua, como Cristina Bañobre y gente de la Universidad, de la Facultad de Ciencias de la Universidad de la República. Hay varias universidades. Yo soy de la Universidad de Viena, también está la Universidad de Texas en Austin, Chief Scientist Brett Baker. Están universidades de California, Santa Bárbara, UC Davis, Harvard. Sí, estamos estudiando microorganismos tanto en la superficie como en los sedimentos. Ahora estamos dando indicaciones de dónde está el Falkor, el barco, con respecto al submarino. Seguimos viendo estrellas, kilómetros y kilómetros de estrellas. Algunos dicen noche estrellada.
I mean, we still have like 7 cores.
Estamos decidiendo cuántos cores vamos a tomar. Saludos. Saludos a todos. Sí, hay muchos Ophiurius. No sé cómo pronunciarlo, Ophiurius. Sí, estamos buscando los orígenes de los eucariotas. Estamos buscando el origen de la complejidad celular o eucariogénesis.
see something cannot stop the camera.
I see. So let's do.
That's the speed. I'm looking at this camera and looking further out.
I see.
What I see here.
Esa es una muy buena pregunta. ROV team, have you ever seen this amount of stars?
They're over here.
Yeah, where in which places?
I'm looking right here.
Okay.
And this camera is looking here. What I see here, if you
Not
see
sure exactly.
something in your
I
right,
think it's in this area.
that's the
In this
best
area,
Yeah.
South
Okay.
Atlantic.
closer. I have time to stop.
Sí, al parecer los chicos de
But this is okay.
ROV me están diciendo que sí han otros sitios aquí en el mar Atlántico Sur han visto esta
I see. So this is the
cantidad de estrellas. Una noche estrellada.
first. So we are now going up.
Ahora estamos ajustando simplemente para ver en dónde nos encontramos y qué tan lejos estamos de la costa. Estamos aproximadamente a unos 202 kilómetros fuera de la costa de Uruguay, a 1000 metros de profundidad. Sí, estamos caracterizando también los nutrientes. Queremos saber la cantidad de materia orgánica que tienen estos sedimentos, la cantidad de azufre, la cantidad de metano. Eso también lo estamos caracterizando. Ya. Buscamos arqueas en todo el mundo. Buscamos arqueas en todo el mundo. Sin embargo, estas específicamente, estas Asgard arquea, se encuentran en sitios donde hay una gran cantidad de materia orgánica. Hemos encontrado muchas Asgard arquea en Guaymas, en el Mar de Cortés, en el Golfo de California, en México. Hemos encontrado una gran diversidad de estas Asgard arquea. Ahora estamos viendo el mapa donde nos encontramos. Hemos estado haciendo el recorrido para que se den una idea del recorrido que está haciendo el submarino. Estamos haciendo un close-up a donde estamos para que se den una idea de dónde estamos viendo todas estas estrellas de mar.
Not a very interesting map, but we are sort of in a different.
Yeah. Lo que vemos es
We are going to articulate this
deeper
canyon, right? So
we go in
like
blue,
yeah, can we expect something
1,000
there?
meters, and then we can give the depth. What?
Around there it's 832.
Yeah. 970 metros es a donde estamos. So I think in a site where we can have a clear area, we can.
From the push boards, you want a clear area from starfish.
Possible.
So while do we have like preference for the site? Like, or is it just
Yeah,
it's
something that it's kind of clear.
So what is here? So, what do we see here? Is it like some? Is it a wreck or?
Estamos decidiendo, y mientras decidimos encontramos estos cangrejitos.
So could it be? So if we find a gold, we definitely take it. So it's really, it's fresh, right?
It can be leftovers of fishermen.
I see.
Sometimes last time we got it some
Sí, estamos viendo
from the fishermen.
algunos botes pesqueros que se quedan
But these crabs,
en el
does it
fondo.
mean they have some something to eat here?
Y vemos 6 cangrejos que se están alimentando de esto. Parece un... It does look like, but from fishermen's. Sí, colaboramos con grupos de exobiología. En particular, yo trabajo mucho con temas de astrobiología.
So these tiny, these small things that these fish
Y características de arqueas y de evolución del metabolismo microbiano y lo que permite a los microorganismos sobrevivir en ambientes extremos. Miranda pregunta qué es lo que he estudiado para poder hacer el océano en profundidad. Estudié la carrera de biología en la UNAM, en México. Después me especialicé en biotecnología, en biología molecular. Hice mi doctorado en el Instituto de Ecología. Hice un doctorado en ecología microbiana y después me especialicé en bioinformática. Y a mí me apasionan los ecosistemas que son análogos a ambientes primitivos donde se originó la vida, estromatolitos, tapetes microbianos, ventilas hidrotermales. Y así es como empecé a estudiar el fondo del mar, en un submarino en el Alvin. He estado a 3.000 a 2.000 metros de profundidad tomando sedimentos marinos, y ahora con el Sebastian. So I think if we kind of go like a little further in this area, here, if we find something that looks,
Just push forward.
yeah, just look a little bit to kind of reach the.
If you see something you want to look close, let me know. Stop and I will
Thank you.
The speed, if you want faster or slower, just let me know.
Yeah, I mean, we need to recover the course, so as
You need to find a clear area.
I think we are there. You know, this is where we are.
this is where we are.
Yeah.
Okay, so let's just find a clear area where we can. And so, if it's possible to fire the listing like a way of any like living organism.
Okay.
And so first we'll do the missing bottles. 4 to just 6. And then we do the push pulls. And if we can get the pH.
The pH readings.
Hmm?
The pH reading is constant, right?
Is it?
She's asking about the pH readings.
So what can we do it now? Like,
Sí, tenemos un secuenciador, un nanopore,
I think we can.
Promethean se
Are you
llama,
guys
es del
bored?
tamaño de una
Just get some
barra de pan.
I think, I don't know what's the order, but
Y como parte de mi proyecto de National Geographic vamos a poder hacer secuenciación en tiempo real y poder tener un análisis de la diversidad microbiana y un análisis de el potencial metabólico de estos sedimentos marinos, además de poder encontrar estas Asgard arquea.
Could focus on she just wants the skin to be clear. So you can fire it and go in, stop and whatever you want to do. You can't stop because I'm going to have to stop the vessel. So it's either you speed up a little or just go to your port side a little. You ready for the next skins?
I think, yeah. Whenever you guys are ready.
All three?
All 3.
All three. Nice.
Okay. So, Robert,
Number 4.
Niskin bottle 4, Niskin bottle 5. You can do only one by one, so you have to do first one.
Sample ID.
Yeah, just do each one.
What is the sample ID?
Yeah, that's all fired.
Yeah. Give me 1 second. Just do each one. Oh,
Good shit,
is
but yeah,
station.
done.
Is station 2, and then S0863. That's the name of the time.
Yeah, nice.
Station 02, and then S0863, and then do 4, and then you copy that one, and then do. You have to do one by one. Submit, and then you do the same
Just
for the other
process.
ones.
Right
Find
now?
out
Yeah, yeah.
position where it doesn't have
It
all
it
right
already
place.
happened, but yeah.
And all the
You
others.
brought it?
So what do I? So what? Break. This
It's
right
supposed to
much.
find a position
So-
where it doesn't have all these stars.
So it's yeah. So it has to be. I think I need to go over the login, but it has to be the time and UTC,
Yeah.
which is the time over there,
Yeah.
and then
But this is for the Niskin, it's here.
Yeah,
So I just wrote the time, right?
it's the time that it's
Yeah.
here, then your the lat long,
Mm-hmm.
and then your initials.
Yeah.
And
So-
any.
We're coring now.
Yeah,
Okay.
we just dropped the fire the skin.
I want to get a estimate on recovery to give people. If we're coring now and we go up, you don't want to
It's
explore
like
it.
2
I'm thinking.
2 hours.
I'm thinking 2.5 to be safe. It's 12:30, so probably
3.
3. I'll say 3 recovery. Does that sound reasonable to y'all?
If we do the coring now.
Say that again?
If we do the coring now,
Yeah.
approximately like 2 hours, 3
To
to
recovery.
recover.
Yeah, you want to recover 3?
Yeah.
Yeah. The coring should just take us like 30 minutes.
30 minutes. Okay. I'll put it as 3 for
Push
recovery
course.
then.
Okay.
Thanks.
So whenever you guys.
So you want it like this?
Yeah.
Because I know you told us you
Yeah.
want it like this.
Yeah, something. I mean, it's not gonna be
We want to make a little bit more.
but maybe just like over there.
Where? Like if there's a spot in the center.
Yeah,
Great. You could come to a slow pause.
like there.
Can we just slice up?
Yeah, just like right there. Yeah, they're gonna move, so.
There's a little spot here.
That is perfect. Robert, can you log pH 7.89? And that's gonna be. No. Environmental observation and then additional text or order because there is no pH 7, 8, 9.
7.89.
No,
Yeah.
any trip. No, let's go.
Estamos decidiendo dónde vamos a hacer el sitio. El muestreo de los siguientes cores porque toma alrededor de 2 horas para subir el submarino y tenemos que estar despiertos para poder procesarlos todos en el momento en el que los obtenemos. Así que va a ser una noche larga. So we have refresh. So yeah, this is the one that we did. So yeah, 7, 8, all the way to 14 if possible.
17 and 18.
Those are the special ones.
Oh, okay, so we need to do those two.
So all? Just up to 14?
Those are the special ones,
These 2 are the special ones.
and then so we're not doing those two.
Okay.
The rest we can.
So what happens?
So those are for
Okay.
So those are like
I see.
the ones that
That's
we need to do for sure.
Okay. 7 and 8.
Oh, this is exciting.
Es otra expedición, no es la misma de ustedes, es otra
Uvidíš, no,
expedición.
oni to přeskočili. Ne.
Empezamos el 11 de noviembre, vamos a terminar el 3 de diciembre y estamos en búsqueda de nuestros
Yeah.
ancestros,
So what is
los
the
Asgards.
what's the number of the
Okay.
core? We are
7.
7.
Let's see. 7 or 8. We're gonna use 7 and 8.
Okay.
Vidíš
noviny?
Sí, 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 todos los que siguen, sí.
Oh, ještě ne, ale je to 8. Teď to tam je číslo na to napsaný.
Yeah, we're not logging until we tell you. So first we're gonna put them in the floor, sea floor, and then once we know and that they look good and they look good, once they go into this white river, that's when you push, hit the sample, hit the button.
It's only like so.
Huh?
The camera.
What I'm saying?
It's only like so.
Can we put first all of them and then we recover?
Oh,
Yeah. Is that possible? That we just get an idea of how distributed they are
Yeah.
and then we recover them later? So we just want to have a.
View of the arrangement.
Yeah. Thank you.
Thanks, Val, for doing it, you know.
So, I know this one is the last one. Do you want me to just do it and put it back? Because I have to use the other arm for the next side.
Why is the last one?
Because
This
the
one's the last one on
this
this side.
They are in different positions, it's different arms.
Yeah, yeah, yeah.
What he's asking is
Oh.
because they are not like before.
Okay, let
on
me
the same bundle.
write that down. Yeah, it's okay. As long as I know that this is in the second station.
Yeah, because we use different arms.
Yeah, 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 yeah. So on the notes we're gonna put 1 to 6 on the first side and then 7 to 14 and 11 on the second side. And then we have the coordinates here is 3, 6,
29.
3, 6, I can't see. Wait. 1, 6, 6, and then minus 53,
23, 36.
46. Second side. And so you put that
So I
Do
put
you want to do the specials?
in here. Or I did it recorded.
We're gonna do the specials now.
Okay.
Robina, teď jsme dělali 8 a 7, nebo co jsme dělali? Teď se dělalo 7 a 8. Jako 2 se dělaly, teďko ne.
Ready?
Buenas noches a todos los que nos están sintonizando. Estamos en nuestro segundo sitio de muestreo. So push core 17 on the quiver, Robert. 17. Vimos muchas especies al inicio de la transmisión. Si quieren ver el inicio de la transmisión, vimos calamar fresa. Estuvo muy bonito y vimos unos tenóforos también. You did already 18?
No.
Okay. O2 is low because we are almost 1,000 meter depth, and it's expected that oxygen concentration will be low at this depth.
This is cool.
Claude, if you can put all of them and then we retrieve them at the end.
That is the last one on this
This is
side.
the last one on the side.
That's the last one on the side. Perfect.
Yeah.
Because then we need the other arms.
Oh, from the other arm. Okay. Got it.
So the other side I'll give you what
Yeah.
you want.
Yeah. Okay, Robert, push core 18.
Okay.
Estamos recolectando también agua. Agua y a esta profundidad, sedimento y agua en la superficie también. Push core 18. It's on the quiver. And then we're going yeah, so if we put from the other arm, all of them kind of distributed. Mi nombre es Valerie de Anda y soy científica de la Universidad de Viena, en Austria. Y soy exploradora de National Geographic también. Soy mexicana.
is our studying oceanographic. Yes, I was trying to explain to you. That was studying. But I mean, yeah, it should be.
No, no soy estudiante,
It's possible.
soy científica investigadora. Yeah, so we have now, we're not gonna do all of them. Just, yeah.
Yeah, you showed us.
Yeah. If we just put them first and then we do the, how do you call this? The waypoint, just to have
The arrangement altogether.
the arrangement. Yeah, exactly. Sí, soy Valerie de Anda con IE al final, no con Y.
So we do already cut us on
Sí, soy mexicana de la Ciudad de México.
Can you wrap out your camera? Wrap out your camera.
So for the notes model,
Camera. Can you rack it back out?
we need to
You know,
just specify we did 2 sites. The first site was push core 1 to 6 plus core 11 that was Jacob's. And then this on the second site, I have it also in my notes, so we can
okay,
just
yeah, just something easier.
yeah. And then this is the second site. So
So-
we just need to kind of get the coordinates, pH, and then once we have kind of the distribution of all of them, Claude just did 17 and 18, and we're gonna do the ones on the right arm, which are 9 to 14. We don't need 15 or 16.
Oh, you don't need a 15, 16.
Sorry?
Solo hasta los 14.
Sí.
So why do we do it like this? Like, why don't we do it like one by one? Just ask it, you know.
Why what?
She want to get the picture of the arrangement. The
Yeah, we
array.
just want to get the picture of the arrangement, because
Yeah, that
Yeah.
way, understand.
some of them are paired and we want to get them as close as possible, especially for the oxygen profile that Jacob and Emily are doing. So this way, just get all of them. Awesome.
Did you need a picture or anything? You're good.
Sorry?
You're good now? This is fine?
Yeah, so we have 9, 10, 11, 13, 14, so we have
11 is done. No?
oh yeah, 11 is done. That's correct. Yeah. That is
Okay.
perfect. So can we just put the drop the waypoint?
Want a waypoint here?
Yeah. Awesome. So now, Pavel, the location that you see over there,
Yeah.
that's the one that we record for the second site.
Okay.
So it's 36 minus 3690 29 16. So that's the one. So yeah, basically we want to do this for all the reporting. Even if we forget, we want to do waypoint, get the coordinates, get the distribution of the course. If there's one site, we put all of them in one site. But if, like now, we did 2 sites, we just want to keep it simple. Están preguntando sobre Asgard. Sí, estas arqueas primero se descubrieron en el Océano Atlántico norte, entre Groenlandia y Noruega. Y se encontraron en una ventila hidrotermal que tenía forma de los cuernos de Loki. Y a partir de ahí se empezó la nomenclatura. Bueno, los nombres de Loki, Thor, como lo sabrán de algunas películas de Marvel. Incluso se le llamaron en las noticias microbios Marvel. Hemos nombrado bastantes arqueas dentro de este grupo, y la más importante es, para nosotros desde un punto de vista evolutivo, es las arqueas hot, que son las más cercanas a los eucariontes. Okay, so whenever did you get the coordinates, Pavel, from this site?
Do you need the whole thing, like the, or just the?
Just lat and long. Yeah.
We need it. Look at it.
Yeah. Let me get a little closer. Yeah. Cell phone. No, just my phone is here. Thank you. Yeah. Awesome. Beautiful. So, Robert, whenever, push core. On 14.
14.
go up and down like a whirlwind range. How did the submarine goes up and down? Like, what's the mechanism?
The thrusters. Up and down.
Para aquellos que nos ven, estamos terminando el muestreo, estamos terminando estos últimos 3 cores y una vez que los recuperemos, el submarino tardará aproximadamente unas 2 horas, hora y media en subir a la superficie, y en cuanto suban tenemos que estar listos para poder procesarlos en el puerto frío. Todo esto se hace porque necesitamos bajar, digamos, un poco las actividades metabólicas de estos microorganismos y queremos que se mantengan a una temperatura baja para poder hacer procesamiento después. Push core number 12. Robert?
Sorry.
Okay.
Ah.
And 2 more. One more left.
Like just straight up now. Trying to decide.
Hmm?
This sounds now. Go up now. You want to explore? Probably should just.
I
go up.
think, yeah.
Okay.
Number 10, you got that one?
This.
Estos sedimentos van a ser procesados en la Universidad de la República de Uruguay. Van a ser procesados aquí, a bordo del Falkor. Vamos a hacer secuenciación en tiempo real. Mi estudiante Swati ya está terminando de hacer la extracción de DNA de los sedimentos que recogimos en la mañana. Push core number 9. You got that one, Robert? That's the last one. Sí, va a haber transmisiones durante toda la campaña hasta el 2 de diciembre, así que va a haber tiempo de que nos sintonicen. Sí, usamos nitrógeno líquido y tenemos refcos que están a menos 80 grados. Ok.
¿A dónde vamos, Linda? Seguimos el mismo camino, we keep the same path.
Sorry?
We keep straight up.
I think we can go whenever. We can go up whenever.
The same path we were following before, northwest.
No, I think she's talking about recovery.
No, recovery.
Oh, you can recover now.
Yeah.
Okay.
How long does it gonna take?
1 hour. start with now. Confused. 3 o'clock too, but okay. I need to do end of time stuff.
I mean, if we can explore a little bit and we can do it at 3, it's fine, but we can go off. If that works for you, we can
Yeah,
go
I mean,
off.
It's just fine. You just need a few minutes to prepare.
Yeah.
That's okay,
No.
so that knowing in advance is
Yeah.
good in communications. Because we also then need to make sure that tech department is prepared. So we're covering
It's
people
here.
and,
Yeah.
whenever you are.
Yeah.
Start from up.
Yeah.
Okay, coming up.
It's like so okay. So how
Very just RV But right there.
Do you want to say a final words to end this?
Yeah, that's RV what's up?
Gracias a todos los que se conectaron en esta transmisión. Hemos recolectado las muestras que necesitábamos, así que vamos a proceder a subir a la superficie. Tomará aproximadamente 1 hora para poder
so Mike,
subir.
how long does it take to go?
Y pues muchas gracias a todos. Esperamos verlos en la siguiente transmisión. Estaremos mostrando más acerca de esta expedición en búsqueda de las Asgard arqueas. Y gracias a todos por conectarse. Les mandamos muchos saludos y les pedimos que sigan haciendo preguntas. Trataremos de hacer lo mejor posible para contestar tanto en inglés como en español. El equipo de la expedición, Chief Scientist Brett Baker, Pavel y Robert de la Universidad de República Checa y yo de la Universidad de Viena y el equipo de team de Sebastián. Les mandamos muchos saludos y gracias a todos por conectarse.